Welcome back to the third module of the NIMAR program. We are now going to look at the HIV life cycle and disease progression. This is the outline. We'll look at the HIV life cycle, then look at how HIV progresses by observing the relationship between the CD4 count and the viral load. Then later on, we will look at clinical disease progression. This is a schematic presentation of the HIV life cycle. You have the HIV virus in green there, and you have a CD4 cell, T helper cell, in a light pink color, right? What you see here is actually an HIV virus that infects a certain CD4 cell receptor. There's like 2, 3, 4 important processes that happen leading to the budding of baby viruses that would then mature and restart the process and infect more cells. So every time HIV replicates, you get more viruses, but then you get the destruction of our CD4 T cells, right? So the HIV virus is a virus that has a bilipid membrane, right? It also has a nuclear capsid in blue there. Inside the capsid, you find 2 strands of RNA, right? Remember, we are DNA species. HIV is an RNA virus together with other enzymes, which we will talk about later on. So HIV would then approach a CD4 cell or a cell that expresses CD4 receptors on its membrane, right? In order for HIV to infect a cell, there is like 5 key important steps you need to know at the NIMAT level. The first step is attachment, right? This is where we get certain proteins, glycoproteins that you find on the membrane of the virus, GP120, GP41, that would then attach to a CD4 receptor, right? But for attachment to be completed, there should be attachment at the CD4 receptor, but also attachment at one of the core receptors, right? So you've got the main receptor and 2 core receptors. An HIV virus would then choose whether to attach to a CCR5 or a CXCR4 core receptor. The majority of HIV viruses prefer a CCR5 core receptor. As soon as attachment is completed between the glycoproteins and a CD4 receptor and its core receptor CCR5, we then say attachment has been done, which then leads to the second step in HIV infection. That step is what we call fusion. Fusion means now the 2 membranes, the membrane of the virus and the membrane of the CD4 cell, fuse or join together, right, to allow direct communication between the cytoplasm of the HIV virus and the cytoplasm of our cell, right? So the second step of HIV replication is fusion. Once fusion has happened, you've got the release of the genetic material, right? The viral genome is released into the cytoplasm of our cell, which is the 2 RNA strands and some of the enzymes, which I'm about to talk about, right? The first enzyme you need to know is what we call reverse transcriptase. It is called reverse because it will translate a single strand RNA into DNA, right? So what is the function of reverse transcriptase? It is to translate, it is to decode, it is to convert a single strand RNA into a double strand DNA, right? Once the DNA is formed, it will be transported into the nucleus where the viral genome DNA would be integrated into the host DNA, which is our DNA, right? The process of insertion is called integration, and it is facilitated by an enzyme called integrase, right? So what is integrase? It is an enzyme that facilitates the insertion of the viral DNA into the host DNA, which is our DNA, right? Once this process happens, basically lifelong infection has been established, because now this DNA, which has the code for the virus, will then be translated during mitosis. Sorry about that. During mitosis to then formulate new baby viruses. A process that follows is then the translation of this DNA into messenger RNA, leading to the formation of amino acids and new proteins. Now, some of the proteins that are formed, these are multi-chain polyproteins, right? Long strands of multi-chain or polyprotein chains, right? That need to be cleaved. They need to be cleaved such that they allow the formation of new viruses, but also important for the maturation of these baby viruses such that they can then complete the cycle and go and infect more cells and restart, right? So the cleaving or the cutting of these long proteins, right, polyprotein chains, is facilitated by an enzyme called protease, right? Protease. So if you look at the enzymes we have mentioned reverse transcriptase, we have mentioned integrase, we have mentioned protease, which cleaves or cuts long strands of proteins into short ones, allowing for maturation and formation of new baby viruses, right? So the critical stages of the HIV life cycle are attachment, right, fusion, right, reverse transcription, right, integration, right, and then cleaving and cutting of long polyprotein chains by protease, right? Just note that these stages also start to give us some ideas around how ARVs work. Target points of ARVs, we've got drugs that inhibit attachment. They are called entry inhibitors. We've got drugs that block fusion, and they are called fusion inhibitors. Fusion is one of them. We've got drugs that block this enzyme, reverse transcriptase, right? These are called reverse transcriptase inhibitors, right? We've got those that are nucleoside and those that are non-nucleoside. So just remember this. So non-nucleoside reverse transcriptase inhibitors is drugs like nevirapine and efavirenz. There's second generation NNRTIs which are beyond the scope of this workshop. We've got nucleoside reverse transcriptase inhibitors. This would be drugs like zidovudine, lamivudine, abacavir, tenofovir, and so on. Then you've got integrase inhibitors, which is a new class which has recently been added to our guidelines. The main one that you need to know is dolutegravir, right? There's others, but for the South African context, we are only using one of those, right? Then we've got drugs that are protease inhibitors, like lopinavir, ritonavir, which is the main drug that we use as part of our first line or second line in South Africa. It's a combination of two drugs, a formulation that has two drugs, lopinavir and ritonavir. We are still going to do the drugs later on, but for now, know the HIV life cycle: attachment, fusion, reverse transcription, integration, and then cleaving and cutting of long polyprotein chains. Here are some questions to reflect on. Please do submit your answer to these questions so that we can then proceed going forward. Thank you very much.